of the human body. Okay, normal flora of the human body. Bacteria which are normally present in the human body and most of which basically do not cause any of the disease in human beings. And what are the basis of the normal human flora? Okay, for example, if there were no bacteria, we would not be able to digest our food. Okay, we would not be able to digest our food. Okay, so the normal flora helps us in a variety of ways. Okay, helps us in a variety of ways. So in our body, humans are born without bacteria, but our bodies are quickly inhabited by hundreds of species. Okay, although we often think of bacteria as evil creatures, threat makers, etc., etc., but in actual scenario, most of the bacteria are actually harmless. Okay, most of the bacteria. Okay, for example, most of the bacteria in our body can be found in your gut, which help to digest your food or even break down. Okay, but they also have an embarrassing side effect. Okay, if you one may notice, okay, when you eat beans. Okay, for example, another example, in a stomach where food is broken down and further in your stomach, okay, resulting group is passed to the small intestine and muscles in your throat push food into your stomach, mouth, etc., etc. So basically bacteria are involved in each and every step of your food digestion. Okay, they helps us to digest food. Some of the essential vitamins are basically the products of the bacteria. Okay, this is how a bacteria are going to help the normal of the human body. Okay, so moving forward with the further digestion. Okay, in the small intestine, okay, special snippers called enzymes cut up long molecules called enzymes, and your body can't use the shorter ones that it can, but the enzymes doesn't work with the big molecules called oligosaccharides, which are found in the some food such as beans. In the large intestine, the oligosaccharides passed to your large intestine where the bacteria can break them down as they do, and the bacteria also produce hydrogen and methane gas, which builds up small amount of fat from it. Okay, so there are variety of functions of the bacteria. This one is a small video of a normal flora. Okay, please have a look.
Take a close look at your hands. A real close look. See all those microscopic critters? Your skin is home to around 1,000 species of bacteria that make up your skin flora. Are they all bad? Do they do anything more than just make you sick? Or do we need bacteria as much as they need us? Bacteria start colonizing your body the moment you're born. You can't get rid of them, no matter how many times a day you take a shower. On average, 5 million bacteria cover every square centimeter of your skin. But don't stress out, all those billions of skin bacteria combined would fit into a ball the size of a pea. You can't even see them with your naked eye. While some of your skin inhabitants pose serious health problems, most bacteria are not only harmless, but beneficial. So what's good about being a walking zoo, and what kind of animals live there? All those bacteria aren't just taking up residence for nothing. Some of them are helping other important bacteria survive on your body. Others just leave no place for harmful bacteria to settle. There are bacteria that alert your immune system to incoming pathogens. Even though you can count 1,000 different species of bacteria residing on your skin, there are 3 of them that dominate: Propionibacterium, Corynebacterium, and Staphylococcus. Bacteria don't cover your body evenly. Just like we don't build our homes in the coldest parts of the planet, bacteria have their own preferences when it comes to your skin. Oily surfaces, like your face, can be much more densely populated, with up to 80 million bacteria per square centimeter. Those oily areas are called sebaceous zones, and they're a particular favorite of Propionibacterium acnes. Yeah, the ones that cause that annoying skin condition. These bacteria live in your hair follicles and the pores of your skin, but they don't normally cause any problems. Even if your skin looks perfect, it's still crawling with Propionibacterium acnes. They live deep in your pores and feed on sebum, the oily secretion that moisturizes and protects your skin and hair. But once your body begins producing too much sebum, these bacteria proliferate, clogging your pores and causing inflammation. The genus Staphylococcus counts over 40 species of bacteria. The most common one is Staphylococcus epidermis. These bacteria are typically harmless inhabitants of your skin, but can cause infections associated with medical devices like catheters or implanted prostheses. Then, there are the moist zones of your skin, the creases of your elbows, your armpits, and the areas between your toes. They're home to Corynebacteria. Some species of this bacteria can produce toxins and cause diphtheria. However, their non-pathogenic relatives are widely used in the mass production of amino acids. Some species even produce substances similar to antibiotics. It's kind of gross to think about how many bacteria are crawling on your hands, neck, and face while you're living your life, but they're a part of you. There's a whole bacterial civilization using your skin as land. You should feel good about it. For them, you are their whole world. And it's not just your skin that bacteria call home. In fact, half of the cells that make up your body aren't human cells. They're bacterial ones. Want to know more about this human safari? Stay tuned and discover your amazing body with us. Since you're still here, while there are 1,000 types of bacteria covering you from head to toe, there are even more types to be found inside your mouth. Over 1,200 of them. Ugh!
Okay, I'm moving forward with the normal flora. I am moving forward with the normal flora. There has been now greatly refined, okay, such as presence of Heliobacter pylori, okay, Heliobacter pylori, which you all know is basically a pathogen also, okay. So this one is basically a type of fossil which was basically found, okay, also known as Iceman, and from this 5,300 year old European Copper Age mummy, and usually by means of newest methods of DNA sequencing, it was possible to reconstruct genome of Heliobacter pylori preserved in the stomach of the Iceman. And the corpse discovered when melting ice revealed the human remains on the side of the mountain. The Iceman Heliobacter pylori genome sequence turned out to be almost pure representative of the Asian population, which means Heliobacter pylori strain was present in Europe before the hybridization of African and Asian strains produced the modern European variants. Okay, so it has been there for a very long time. Now, what are normal flora and how the term normal flora implies? Okay, the term normal flora implies that these microbes are basically harmless. For most part, they do not cause disease and are even beneficial, and most are commensals. Okay, most are commensals. They benefit from the association with the host, but the host is not affected. And others have a mutualistic association with the host, and they benefit the host in some fashion while thriving in the host body. Okay, it is also important to learn that the beneficial effects and how the beneficial effects are reduced nowadays by use of excessive use of antibiotics also. Okay, by use of excessive use of antibiotics, other than helping the bacteria, helping the human beings with the digestion related works functions, or it may be another functions, they also help us, they also help us to survive against various other pathogens also. Okay, so some flora, normal flora, can be opportunistic pathogens. That is, they may cause infections if tissue injury occurs at site specific, or if the resistance of the body to infection is decreased. For example, E. coli, it's a normal flora of an human body which is found in the intestine. Okay, it does not normally harm our intestine, but due to some or the other reason, if there is any change, if there is any change with the diversity of the normal flora, okay, there are chances that it may infect our intestine, but up to a limited extent. But the main problem lies if the same Escherichia coli enters our respiratory tract. Okay, the same Escherichia coli, if it enters our respiratory tract, okay, then in that scenario the E. coli for us can be fatal to us. Okay, E. coli can be fatal to us. Okay, so the word opportunistic pathogens for some of the normal flora is also used, and this is especially important because in recent years there has been rising incidence of infections from these microorganisms, but the main reason is behind the lifestyle of a human being. Okay, and the change in the pattern of the food also, and also the overuse of antibiotics or the overuse of any chemicals which basically limits the growth of the human microbial flora. For example, nowadays you already know, due to COVID we all are using uh, sanitizers, and sanitizers, it may kill virus, but the problem is it also kills our normal flora of the human skin. Okay, normal flora of the human skin. Okay, in that scenario, in a longer term, we may have problems related to fungal infections also in our skin due to the decrease in the normal bacteria which are normally present in the human skin. But the problem lies with the overuse. Okay, with the overuse of the sanitizer. For example, if you use every hour, every 5 minutes, whatever, okay, if you use very frequently, okay, there are chances that there will be decrease in the, okay, at every age point there will be a difference in the composition of the normal flora, and there are also various other factors such as geographical conditions. Okay, and the conditions which are basically around you, okay, will also affect the composition of the normal flora. Okay, will also affect the composition of the normal flora. Okay, now the normal flora, and how the concept of normal flora came, okay, was devised basically, and what effect does the establishment of normal flora have on the body. Okay, so there has been three approaches to answer this question. First one is the use of germ-free animals. Second is basically use of antimicrobial agents. Third one is the knowledge of certain characteristics of the normal flora organisms. Okay, so these are the approaches which have been used, okay, for the study of normal flora. The concept of germ-free animals. Okay, this basically includes by growing a animal without a single microorganism in it. Okay, without a single microorganism in it. Okay, but we are not going to grow human beings as such. Okay, we are not going to grow human beings. If the colonization of the experimental animals by microorganisms can be prevented, and one can compare the properties of such germ-free animals with those of the normal animals, and the results can be helpful in understanding the functions of the normal flora of the human beings. Okay, normal flora of the human beings. Okay, so what we are going to do, okay, we are going to avoid the colonization of an animal during its birth. Okay, we are going to avoid colonization of microorganisms in the animals during the birth of the animals. Okay, second is basically use of antimicrobial agents. Okay, and what effect does it have on the normal flora? If the balance that occurs between normal flora and human host is altered by use of such agents, various effects may also occur that are useful for indicating the role of normal flora of the human body. Similarly to the previous one, again we are not going to use human body, we are going to use experimental animals to find out what are the roles of normal flora in the human body. Third one is the knowledge of certain characteristics of the normal flora. Few years back, for example, at least 20 years back, okay, there was no proper procedure available to study normal flora. But nowadays, a very high quality research is going is under process to study normal flora by using molecular biology and there are variety of projects which basically support the funding of to study the normal flora of the healthy human host. Okay, to study the normal flora of the healthy human host. Now the concept of germ free and the gnotobiotic life. Okay, and gnotobiotic life, this concept is the basis which made the basis of the normal flora. Okay, earlier Pasteur did not believe that animals could live in the absence of microorganisms, and in the year 1897, following his suggestion, an attempt was made to rear germ-free chickens, and between year 1899 and 1908, Scuttulus, a German, was successful in raising chickens that were bacteria free. However, because his birds did not develop normally and died in about a month, he concluded that the intestinal bacteria are essential in the nutrition of the vertebrates. So, when they made their first trial with the chickens, they found that the chickens, okay, which within a time span of 1 month from their birth, almost all of the chickens died. Okay, so he concluded that the intestinal bacteria are one of the essential in the nutrition. Similarly, another trial was done in the year 1912. Kahandi at the Pasteur Institute basically raised 17 chickens for 40 days and concluded that the vertebrate life is possible in the absence of microorganisms. Okay, they concluded that it is possible to live without microbes. Okay, but the problem is with the nutritional requirement. Okay, we know how that an adequate diet is provided and germ-free chickens live long healthy lives and reproduces as well as the normal birds. The problem was rearing of the animals was basically led to some of the conclusions also. One such conclusion was they needed high doses of vitamins also. Okay, so in the year 1928, James Skinner at the University of Notre Dame started work on the germ-free chickens. He and his associates developed the equipment and techniques for rearing chickens, rats, mice and other animals in the absence of detectable living organisms for the several generations. They emphasized the anatomical and physiological descriptions of those animals and made comparisons with the conventional and non-germ free animals of the same species. Okay, this was the first experiment where the results were basically compared. Okay, where the results were compared between a normal flora containing animal and without the normal flora containing animal. As a result of these studies, germ-free animals no longer belong to the realm of biological curiosities but have some practical models for solving the importance in the biology and medicine. And germ-free laboratory units basically, okay, are basically nowadays used to study various other functions also. Now this is an old model, okay, and the newer models are basically same which is seen here. Okay, now this is a germ-free equipment, okay, where animal is kept, okay, and the whole condition is sterile. Even the air which is being used, okay, air which is being used here in this case is also filtered, and the food whichever they are provided is also sterilized, okay, is also sterilized. Okay, now compared when these animals, germ-free animals, were compared with the normal animals, germ-free animals exhibit an underdeveloped immune system, making them susceptible to infection and if subsequently exposed to the pathogenic bacteria. So this was one of the disadvantage, and one of the main disadvantage is the basically lower immunity level, okay, which is basically a lower immunity level in the germ free animals. Okay, if they are exposed to a normal environment, there are chances they may immediately catch up a new disease. Okay, and as I told you, they required higher levels of B vitamins. They required a higher levels of B vitamins than the normal animals, and they require vitamin K, and under the normal conditions where normal flora are present, the animals do not require vitamin K. Okay, animals do not require vitamin K. From these findings, it indicates that the normal flora may make a significant contribution to the vitamin requirement of the host. Okay, from these studies, it was concluded that the gnotobiotic animals basically are susceptible to the pathogens, highly susceptible to the pathogens as compared to the animals animals with the normal flora. Okay, and the another main point is they require higher quantities of vitamins as compared to the animals with the normal flora. Okay, and what are the other uses of germ-free animals? Gnotobiotic techniques, okay, have been used to assess the effect of particular species of organism on a host. Okay, Here the germ-free animal is reared in the presence of one or more non-microbial species to determine the effect of those species on growth and development of the animal or on various physiological processes. Okay. Similarly, one can inoculate a germ-free animal with a known species of microorganisms or a mixture of known species to determine their ability to produce disease or cause pathological or immunological changes in the animals. Okay, for example, nowadays gnotobiotic techniques have helped to elucidate the role of bacteria in the causation of the dental caries, and the effect of antimicrobial agents. The suppression of the normal flora can be done by use of antibiotics. Okay, so in this test scenario, okay, where the effect basically can be studied by using a one antibiotic at a time, okay, but it's just for the experimental purposes. Okay, it's just for the experimental purposes. It can be not used in our regular life. Okay, for instance, the treatment of skin of humans with antibacterial agents such as hexachlorophene results in the suppression of normal gram-positive flora and promotes colonization and clinical infection by gram-negative bacilli and other organisms that cannot normally establish themselves on the skin. Okay. In humans which are not receiving antibiotic therapy, such strains are normally held in the check by normal flora and they usually do not grow in high numbers. Okay, and the normal flora can prevent establishment of the pathogens by means of a competition. Okay, by means of a competition for the nutrients and the formation of inhibitory metabolic products and antibiotics. Okay, this is how normal flora helps. Okay, one of the let me give you one example regarding the harmful effect of antimicrobial agents. Okay, a few years back, it is a case of in a Gujarat where a woman consumed too much of antibiotics without the consultation of a doctor, and which led to suppression of her normal flora in the body. Okay, and which led to internal infections by fungi in her body. Okay, and it was up to such an extent that she had to undergo a treatment for the normal flora as well. Okay, and her case had gone critical because of such critical due to the fungi which infected her internal organs. Even the heart was infected, okay, by the fungi in her body, because in the absence of her normal flora, the body was not able to give a proper immune response. Okay, so characteristics of normal flora: many species of normal flora have the ability to adhere to the surface host epithelial cells and thus have a selective advantage over the microorganisms in the colonization of the host. Okay, so resident flora and transient flora. Okay, what is the difference between normal flora, in which one is a resident flora and the transient flora? Resident flora of the microbes includes a constant population which is always present in our body and cannot be removed permanently. Okay, cannot be removed permanently. Okay, but overuse of antibiotics can also affect resident flora as well. And there are transient flora, okay, which are basically derived from the immediate environment and which consist of some of the pathogenic pathway microbes as well as non-pathogenic microbes also, and these are basically found in the skin or mucous membranes, and which may be able to grow, okay, on such surfaces for hours, days, or even weeks, and these are purely derived from the environment around a individual. Okay, now what are the advantages of normal flora organisms? First advantage, they suppress the entry of pathogens, produce vitamin K and vitamin B, raise overall immune status of the host against pathogens having a related or shared antigens, and the antibiotic substance produced by some, for example colicins, have a harmful effect on the pathogens. Okay, and endotoxins liberated by flora may help in the defense mechanism of the body as well against the other pathogens which basically enter the body. There are also disadvantages. They may become pathogenic when the immunity is lowered, and act as pathogen in tissues outside their habitat. For example, Escherichia coli, their usual habitat is the intestine, but when they are, or some of the other reason, if they enter respiratory system, it may have a pathogenic effect also. Okay, another example is given also here. A normal flora of intestine may cause urinary tract infections, okay, which is a different system from the intestinal systems, and cause confusion in diagnosis due to their ubiquitous presence and their resemblance to some of the pathogens also. Sometimes there is a confusion in detection also. Okay, now moving forward with the innate and environmental factors that impact diversity and stability of human microbial flora. Okay, and the table is basically very important here. Okay, table given here, okay, it's very important to understand how normal flora have effects on the lifespan of a human body. Okay, the first one, okay, I have covered each and every point at the age wise. Okay, the age wise. Okay, first one, at the birth of an individual. Okay, at the birth of an individual, innate factors is basically which are going to play in the role of his immunity is basically genetics, and genetics which is basically acquired from the mother and father. Okay, if they are resistant to if they are resistant to the particular pathogen or sensitive to any of the pathogen, it may have effect on their child also. Okay, and the environmental factors basically are caregivers, the persons which are involved in care of a child, for example mother, father, grandfather, uncle, or his elder sister or brother, whoever it is. Okay, and breast milk. Okay, and immunizations such as use of vaccines also. Okay, which these are the environmental factors which are going to affect the normal flora. So starting with the diversity, at this point, okay, around the time of the birth, microbial diversity is generally low. Okay, or you can say almost zero also, and as there is no diversity, there will be no stability as well. Okay, and one of the most important rule of the nature, Okay, you all need to remember, in any of the living environmental systems, higher the diversity, healthy is the nature's system. Okay, the same thing applies to the normal flora also. Okay, higher the diversity, I am not using the word populations, I am using the word diversity. Higher the diversity of the normal flora, okay, higher will be the stability, okay, and have a more of a positive effect also, okay, and have a more of a positive effect also. Okay, now moving forward with the infant stage, okay, innate factors, immunity will start developing, okay, and the environmental factors which basically will add will be, now the child will be able to eat foods, will be able to crawl and come in contact with other persons, as well as may have infections also, as well as may use antibiotics in the case of infections also. So a wide variety of environmental factors will be involved in the first scenario, which will lead to increase in the biodiversity of the, sorry, not the biodiversity but the microbial diversity of the human being, but still there will be no microbial stability as the diversity is still low. Okay, diversity is still low. Now comes the child. Okay, innate factors will be body growth. Okay, now you will see a change in the behavior with, as well as their diet. Okay, and what he does with his friends, okay, or any of the relatives. Okay, and from the contact and a variety of contacts he is exposed through, or he may undergo some uh, some time of playful activities. Okay, some of the adventure activities if he does. Okay, whatever it may be. Okay, but as he is exposed to more number of microbes, now again the microbial diversity will increase. Okay, will increase, and the stability will also increase. Okay, now comes the teen stage, where due to puberty and the sexual activity basically starts in a human being. Okay, now comes the other environmental factors. Okay, previous ones are also main one, but new factors are basically, he will travel, sports, fitness. Okay, and various other interminate bodily contacts also. Okay, and again with the con- with the contact with the other environmental factors also, again the diversity will increase and the stability will also increase at its maximum level. Okay, now moving with the adult stage. Okay, various innate factors which are normally seen is a weight gain, pregnancy in woman, metabolic disease if he has, and also stress like conditions. For example, he is doing job, or he has owns a business, whatever it may be. Okay, and the environmental factors which are going to affect is basically stable cohabitation, job changes, work, relocation, children's medications, or any various other environmental factors. It has been proved that the environment around an individual of a human being has an effect on the normal flora of the human body. But at this period also, the diversity of the microbes will be maximum and stability also will be maximum. Now moving towards a senior stage. Okay, senior citizen stage, there will be problem in the muscles, atrophy will occur, there will be decline in the immunity. Okay, which may lead to caring side, also lead to diet change, menopause, and also lead to declining in the fitness. Okay, and still you can see the diversity is same, but the stability has decreased, but the microbial stability has decreased, which is seen in almost all of the almost all of the human individuals. And in the senescent stage, which is also called the last stage of the human being's age life, okay, in which basically system failures occur, in which basically systems failures occur, and due to the loss of the motility, incontinence, and cardiopulmonary disorders, and various other age related infections also. Okay, again in this case there will be lower stability of microbes, and still the microbial diversity will be maintained. Okay, but the main problem is the diversity will change. Okay, diversity of the microbes will change in these scenarios. Okay, now these are graph which in which the sites which have been sampled under the human microbiome projects. This is just an overview, okay, where they have sampled from the skin, saliva, urinogenital tract, and the gastrointestinal tract. Okay, so in the skin you can see, and yes, this diversity was evaluated using 16 srRNA gene sequence analysis. Okay, and the skin was basically populated with the Propionibacterium species, okay, urinogenital by Lactobacillus species, and gastrointestinal by the bacterioid species, and saliva by Streptococcus species. Okay, so in the case of a skin, where you can clearly see more than half are basically Propionibacteriums. Okay, more than half are almost Propionibacteriums. And second one is Staphylococcus, then comes Corynebacterium, then comes Anaerococcus, then comes Streptococcus, and the last one is a whichever other bacteria which are studied. Okay, other groups, as there are more than 1000 species. You may imagine the diversity, and most of them, okay, cannot be studied because of the limitations of their growth outside the human body. In the case of saliva, Streptococcus, next comes the Pasteurellacea family, next comes Prevotella, next comes Veillonella, next Fusobacterium, next Neisseria, next Actinomyces, next Porphyromonas, and the rest are other bacteria. And in the case of urinogenital tract, Lactobacillus is one of the common ones, but it depends on a male and female also. Okay, the composition depends on the male and female ones also. Okay, next comes Prevotella, next comes Gardnerella, next Atophobium, next Cynthia, next Bifidobacterium, next Megasphaera, next Anaerococcus, and rest are the other ones. You can see the population of the Lactobacillus, which is very high as compared to the other bacteria. Okay, now comes the gastrointestinal tract, where the maximum population is of Bacteroidetes, which is a group. Okay, and next comes a Firmicutes group, next comes Lentispirae, next comes a Proteobacteria group, next comes Standardid groups, next comes Microbia, and rest are the other ones. Okay, where you can clearly see the word in this case word group is used. Okay, the group is used because there are very wide variety of microbes present in the gastrointestinal tract. Okay, So thank you. I end my lecture here. Okay, everyone please comment present sir for your attendance. Okay, and join the WebEx session of 8:40.